In this section, we will discuss the structure of the genome and virion, binding to host cell, replication cycle in the host cell, cell susceptible to HIV infection. HIV is the member of lentivirus subfamily of retrovirus. All retroviruses contain reverse transcriptase, which is an enzyme that transcripts genomic RNA into DNA. The HIV genome comprises approximately 10 kilobytes. It also has glycoprotein 120, glycoprotein 41, nucleocapsid, RNA, capsid, matrix, and lipid membrane. HIV has long terminal repeats. The protein encoding genes GAG, which is group-specific antigen, PUL, which is reverse transcriptase, and other enzymes such as Protease and Ribonuclease, and ENV, which is integral membrane protein. For lipid membrane envelope, it is viral surface protein that mediates CD4 binding and membrane fusion. GAG and ENV are structural proteins. These additional features that HIV shares with other retrovirus. The HIV genome also contains other regulatory genes for transcription or organization of the late replication cycle virion infectivity factor, which increases infectioncity, regulatory of expression of virion protein, and negative regulatory factor. These genes overlap, that is, they are located in the same segment of RNA molecule. Differential transcription by the protein synthesis mechanism of the host cell yields the various genes products. TAT, REV, NEF, VIF, VPU are genes unique to HIV, which can either enhance or inhibit viral synthesis. Several of these molecules also antagonize cellular defense systems. For example, NEF downregulates MHC class I, and hence helps the virus escape immune detection, while VIF blocks the enzyme APOBIC, which destroys the viral RNA. Moving into further details, VPR, NEF, TAT, and REV are regulatory proteins. VPR is transcription factor. REV is regulator of expression of structural proteins. Other proteins include VIF and VPU. VIF increase infectioncity, while VPU enable budding. The HIV virion is 100 nanometer in diameter. Its outer lipid envelope is studded with 72 spikes derived from glycoprotein 120. The spikes are anchored in the membrane by transmembrane protein glycoprotein 41. The lipid membrane makes HIV especially vulnerable to attack by lipophilic detergents, such as alcohol. Remaining components, HIV has RNA, nucleocapsid, lipid membrane, capsid, and matrix. Now let's discuss binding to host cell. Well, the attachment of viral particle to the surface of the host cell involves two steps. In first step, binding of glycoprotein 120 to second domain of CD4 molecule, and secondary binding to a chemokine receptor after conformational change. So, which cells are susceptible to HIV infection? Well, HIV attacks CD4 plus T cell and other cells of the immune system. This includes cells of monocytic system, such as monocytes, tissue macrophage, and Langerhans cells. It is still unclear whether HIV can infect pluripotent stem cells. Cells of gastrointestinal tract and CNS can be infected. Microglia, astrocytes, oligodendrocytes, and endothelial cells of cerebral blood vessels are cells most commonly infected by HIV. It is unclear whether neurons can be infected. Let's discuss the replication cycle of HIV in host cell. Binding of HIV to CD4 and then membrane fusion. Release of virion contents into cell. Then reverse transcriptase transforms RNA into DNA. Next step is that this viral DNA is integrated in cell genome through integrase. Then protease cause production of viral RNA and proteins. There is also production of integral membrane proteins such as Glycoprotein 41 and Glycoprotein 120. Then there is budding of virus particle. And then there is release. This was all about this section. In our next section, we will talk about the diagnosis and treatment of the HIV virus.